Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speakers in the question and answer window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speakers your questions. Your questions in the question and answer window will only be visible to myself and our speakers. So thank you for joining us today. Our speakers today are Caio Enrique Barros from the National Institute for Bioprocessing Research and Training in Dublin, Ireland. He's a research scientist at NIBRT in the Formulation and Stability Group. His interest in mRNA therapeutics led to studies of lyophilized formulations and the mechanisms of mRNA degradation in solution and solid samples under stress conditions. And James Geiger is a field application scientist from PerkinElmer, where he supports microfluidic characterization of proteins and other biologics. James holds a PhD in biochemistry from Arizona State University. Welcome to you both, and over to you, Caio.
Yeah, thank you for the introduction, Leah. So my name is Caio. I work as a research scientist at the National Institute for Bioprocessing Research and Training, also known as NIBRT. I'm based in Dublin, Ireland, and I work in the Formulation and Stability Lab. So we are focused mainly on the stability of biopharmaceuticals, proteins, and now more recently on mRNA formulations as well. So I will just give a brief background of why our group is so interested in mRNA stability. And of course, the first reason is very obvious. We are going through the COVID-19 pandemic. And with this pandemic, we learned that some mRNA technologies can be very valuable for us in terms of providing us with vaccines. So that was a huge proof of concept. And the fact that it's very quick to make or develop new mRNA vaccines was excellent for us. But before that, some mRNA-based therapies were already being developed, especially in the cancer treatment field. And the perspective is that more and more technologies are going to be developed using mRNA-based formulations. The problem of these formulations is that they are considerably, not stable in mild conditions or room temperature. We probably know that some of the COVID-19 vaccines had to be shipped at minus 80 degrees Celsius and stored at that temperature as well. So that's not great under perspective of under logistics perspective, and also from a chemical perspective. There is a lot of room for improvement here from stability point of view. In terms of analytical tools that we have at our disposal to study the degradation of mRNA or study the stability of mRNA, there are a few methods. There's no really consensus on to which one is the best one. Each one has its advantages and disadvantages. For example, agarose gels and colorimetric quantification are two very simple ones that provide some degree of information, but of course they are not very exact or accurate in what they provide to us. And then there's chromatography methods involving sequencing of RNA, and then microfluidics-based technologies such as the lab chip technology, which is the one that I'm going to be focusing this presentation on. So that's why we are focusing on mRNA formulations and their stability. I will just give a brief context of the history of our research group. So we are very much interested in lyophilized formulation. Lyophilization is also known as freeze drying. So basically we remove water from the formulation in order to make it more stable. So right now, about 50% of biopharmaceutical products in the market are produced in lyophilized form. Most of them are proteins and monoclonal antibodies. And when we're talking about protein formulations being lyophilized, it's very well known that you are always increasing the shelf life of that product by lyophilizing your formulation. So by removing water, you are, in summary, just favoring some interactions between your protein and your excipients, which are usually polymers, sugars, and buffers, sometimes surfactants as well. And by doing that, you are increasing the stability of your protein. So since that's our background, it was quite easy for us to start wondering if lyophilization would also improve the stability of mRNA or mRNA formulations or mRNA vaccine formulations. Now, there are a few aspects to be considered here. The first one, obviously, we want functional mRNA strands after lyophilization. So as much as they can be structurally sound, they also have to maintain their biological activity after freeze drying. Also, we have a very vast knowledge in terms of excipients that can be used for protein formulations. But now we're talking about something completely different. So maybe those sugars and buffers and surfactants are not that useful for mRNA formulations, especially given that the mRNA is always being encapsulated in lipid nanoparticles. So actually, it's the lipid nanoparticle that's sort of facing the external environment where the excipients are. And finally, there is a more fundamental question, which is what is the role of water in the degradation of mRNA? So by removing water, are we actually doing anything? Are we actually improving the stability of the mRNA or not? So that's the first question that we wanted to tackle. And that's sort of the snapshot of our research that I'm going to show here. In terms of methodology, what we did was we prepared a mRNA formulation at a concentration that you would usually see in a vaccine formulation, for example. And we chose mannitol as an excipient at 5%. Then we split the sample into two. One remained in solution, the other one was lyophilized. And then all of the samples were incubated in ovens for the stability assay for 12 hours at two temperature values, so 20 and 50 degrees Celsius. And then every couple of hours, we collected some aliquots and prepared the samples for lab chip runs. Some points to note here, we are using luciferase encoding mRNA, and that's produced by in vitro transcription in our lab. Lyophilization was carried out also in our lab in 2 ml glass vials, and we are using the lab chip GX2 touch system using the high sensitivity RNA assay. So basically our objective here was to assess the extent of degradation of the mRNA by calculating how much of the mRNA peak in the electropherograms is maintained over time. So I'm going to show what a typical run looks like in the lab, on the lab chip using the high sensitivity RNA assay. So there you can see the electropherogram, and on the right there you can see a simulation of what a gel would look like. So we have the ladder, which sort of calibrates our system so we can estimate the size of the mRNA that we're dealing with here. The electropherogram you are seeing there's a part in green. That's what I'm calling main mRNA peak region. And then there's a part in red, which we're calling smear region. So that can be defined using the analysis settings in the software, the lab chip reviewer software. And yes, the user can pretty much define the regions of interest and what sort of parameter that you want to extract from the electropherogram. In this case, I am focusing on the percentage of the total area to estimate how much of the total area of the main mRNA peak is disappearing over time. So obviously, we are assuming here that in the main mRNA peak region, we have intact mRNA molecules or biomolecules, which are then degraded over time under temperature stress conditions to some fragments that should appear in the smear regions since they are smaller than our main intact mRNA. Okay, so after running our samples, I'll show here a few examples of what some stability samples look like. So here I'm comparing two lyophilized samples exposed or incubated at 50 degrees Celsius. One after 2 hours incubation, the other one after 12 hours incubation. On the right, you can see again the gel that is created after the run. And some things that we should emphasize here. So as you can see, the most obvious thing to observe here is that the main mRNA peak is basically disappearing or degrading or shrinking along with time. There is a secondary peak there that is being resolved after 12 hours and actually becomes the most intense peak, whereas the first or the main peak after 12 hours, or after 2 hours, becomes a secondary peak after 12 hours. Also, there's a slight increase in the smear area due to the formation of degradation products. And when we compare these runs with the corresponding samples in solution rather than in lyophilized form, we can then start having some insights on the mechanism by which the mRNA is being degraded. So if you pay attention here, whereas for the lyophilized samples, we have a main peak that is becoming the secondary peak after 10 hours incubation. For the solution sample, that doesn't happen. The main peak remains as the main one after 12 hours, even though there is a secondary peak about to be resolved there after 12 hours. So this distinct degradation pattern is very valuable information for us to start establishing some ideas about how the mRNA is degrading under each type of formulation. So after collecting all of the electropherograms after each time point, we could then draft some degradation profiles of the mRNA. So here on the left, we have the solution formulation. On the right, we have the lyophilized formulations. Again, both incubated at 20 and 50 degrees Celsius. So at the start, of course, we're assuming that we have basically only intact mRNA molecules, whereas at the end, we should have a mix of the intact mRNA plus degraded mRNA as well, and fragments. So some things that were interesting to us here from a research point of view in our group, the samples that were incubated at 20 degrees, both lyophilized and solution, they weren't really degraded over time or over the period of this test. We were expecting some sort of degradation after 12 hours for the solution formulation, which didn't happen. Also, the lyophilized sample was the one that reached the lowest level of intact mRNA after 12 hours. And that's quite surprising to us, given that from our experience with proteins, usually by lyophilizing, you are increasing the stability of your formulation, and it doesn't seem like that's happening here, at least after this one experiment. And also it's interesting to see that the pattern of degradation is different. So the pattern of the electropherograms was already different, as I showed in the previous slide, but also the pattern of the degradation profile is different. So for the solution formulations, you have something that resembles more of a linear decay, whereas for the lyophilized formulation, there's something that looks like a stepwise mechanism. So that was very good information for us to have here. So far, I've been talking about main mRNA peak and how it's decreasing in intensity and area over time. But you can also have a look at the fragments or the smear area that you define in the software as another way of looking at the same data. So that's a zoom of the smear area. So you can see that after 12 hours or at the end of the stability assay, there's a slight increase in the area of your smear. And on the right there, you can see a graph showing the increase in percentage of the smear area after 12 hours of our stability assay. So again, that confirms what we already knew. Only the samples incubated at 50 degrees, both lyophilized and solution samples, almost to the same degree, induce the increase in the smear area of the electropherograms, which didn't happen to the samples incubated at 20 degrees. So that's just a snapshot of our research and what we've been doing in our lab, and how the lab chip is being extremely useful for us, for our purposes. So in terms of conclusion, we can say that the lab chip can be used very well to establish a degradation profile of mRNA formulations. In our case, we could go as low as 10 nanograms per microliter, sometimes even lower, which is great because these formulations are often very diluted. And each run takes only a couple of minutes, which is also very positive for us. Also, the resolution was a good surprise. This type of resolution you wouldn't be able to get from a simple agarose gel, for example. So just by looking at the electropherograms, we were able to start establishing some mechanisms of degradation for the mRNA formulations, which was very good for us. And our first research question there, which was, is water doing anything for the stability of the mRNA? And what we saw was that by lyophilizing the mRNA strand, you are actually not improving the stability of the formulation, but there is definitely a change in the degradation profile and probably a change in the mechanisms of degradation of the mRNA. So obviously here I was talking about naked mRNA, so not encapsulated. Right now, our lab is focused on looking at the stability of encapsulated mRNA, which is definitely more relevant since all of the mRNA formulations involve lipid nanoparticles. So that's already ongoing. And again, the lab chip is being able to give us some answers about that. And also the fact that the lab chip has this high throughput character, and the fact that it's very, each sample takes only really 2 minutes to run. It's very easy to screen a variety of different buffer excipient combinations, and that's very important for mRNA LNP stability. So we are also doing that currently in our lab. So that's me. I'm going to, well, James is going to take over the presentation now.
All right, thank you, Kaio. Make sure I can control these slides. So first, I do want to say thank you for all that data that Kaio presented. It's great to see. It's really great to see how the lab just being used in the field and see how it's allowing really cool research to be performed. What I want to do is highlight just some extra publications and give a little more information into what the lab chip is, how the system works, and some other type of data similar to what Kyle has shown. Firstly, on these slides here, we see a few publications here where the lab chip is being used for similar analysis of integrity, amount of RNA in different samples. These are just from the last couple years, and we can see that it's becoming pretty standard in the industry to use the lab chip for this type of analysis. So we have, again, as Kyle mentioned, a couple publications here having to do with COVID-19, an expression of those proteins in different cell tissues, or where the RNAs are going after infection. In a lot of these publications, we don't get data out of this because they're doing qPCR on large libraries and things of that nature. And so we don't actually see the lab chip data here, so I don't have nice, beautiful electropherograms from each of these publications, but the lab chip is being used in each of these to analyze concentration, the purity, the integrity of RNA and DNA. In workflows such as this, it's very, very common before doing RT-PCR or even next generation sequencing for DNA libraries as well. And so we can see that it is becoming pretty standard in the industry to use this technology. And so again, we're super grateful and thankful to hear about this from Kaio and see way more detail into how this is being used. With that in mind, I'm going to show a couple other applications and just highlight kind of how the system operates so we can get a good idea of what to expect if we were to use this. The first application I'm just going to talk about briefly is looking at modifications of nucleic acids on the lab chip. So this is from a white paper that we put out as PerkinElmer a few years back. But essentially, when working with nucleic acids, DNA or RNA, sometimes we want to modify them in some way, and this is going to help us achieve our research goals. So there's a few that are highlighted in this white paper. First is RNA capping, phosphorylation of DNA, or adenylation of DNA. Essentially, we're doing enzymatic mediated modifications of these nucleic acids. And something that the lab chip is pretty useful for as well is being able to measure the efficiency of these reactions and to determine the percent population of your sample that this reaction has occurred in. So how much phosphorylation has occurred? Is 50% of our DNA phosphorylated, 75%, so on and so forth. And so we're already pretty familiar with the electropherograms from what we've already seen today. But here we have a few examples of quantifying these types of reactions. You see on the left there, we're looking at RNA capping. These is the same fluorescence traces versus size that Kaio has shown. And we have a negative control on the bottom looking at no cap RNA. And then we have a positive control on the top where we see a known mixture of the capped versus the non-capped RNA samples. And then as we move up the electropherogram on those three traces from the red to the green, we titrate in more enzyme. And as we titrate that enzyme in, we get a higher efficiency of this reaction. So you can imagine in a bunch of different RNA samples or with different enzymes or different formulations, we may get differing amounts of these peaks. And so it's just to show that the lab chip is able to pick up these small changes and modifications on this RNA. And then also we can look at reaction efficiency as we pick up these changes. We see a similar thing for DNA phosphorylation over on the right, where we have, again, our controls. And then as we phosphorylate more and more, as we move up, we see these shifts in size. And we can quantify the amounts of each of these and to figure out exactly how much, 70%, 20%, so on and so forth. What does the population of these nucleic acids look like? And again, as we've already heard, in very quick format, right? And so this allows us to be really high throughput and measure a large population of samples in a limited amount of time. With that being said, I'm going to change a little bit what I'm talking about and just give a bit of an overview on the system itself. So everyone has a good idea of kind of what it is and how it works. The lab chip instrument is over on the right of the screen that we're seeing right now. This is a fully installed Windows 10 PC on the instrument itself, and we have a touchscreen on there. That allows a small footprint on the lab, so we're taking up about maybe a 2 foot by 2 foot space on a benchtop, which is really nice. We're not invading 20 feet of bench space in a small lab already. But what the real hero is of this is the chip. So this is a capillary electrophoresis system. So that chip that we see in the middle of the screen is a quartz chip with capillaries literally scratched into that. And so this is what allows for the very, very quick analysis. Where we have, roughly 60 seconds per sample. Different assays have slightly different times, but around a minute per sample is what we can expect. And what allows us to do the really high throughput analysis as well. These chips are multi-use, so they're validated for 400 for protein, 2000 for DNA, RNA, and you can use those every day for as long as you'd like. And what makes the system awesome, I think, is that it's validated with ready-to-use assays. So for each assay that we want to run, we have a reagent kit like we see towards the top of the screen, and then we have a chip in the middle. And the reagent kit comes with pretty much everything you need to run the assay besides your input, your samples, the gel, the dye, things of that nature. And these assays are very plug and play. Take the gel, put it in the chip, load the chip into the instrument, and you're pretty much off to the races. The types of metrics that are commonly analyzed on this lab chip, as we've kind of already seen a little bit today, titration, so looking at concentration, looking at purity of different peaks, stability or integrity, like we've seen today. We have glycosylation profiling assays, charge variant assays, and we've already seen some of our data looking at enzymatic reactions and how efficient these reactions are happening. All of these metrics are able to be calculated for both the RNA that we've seen today, as well as DNA, and then we have protein assays as well. And so this capillary electrophoresis instrument is very, very quick. And I want to make sure that by the time we're done today, we all have a good kind of idea of how this works. And so we're not just looking at magic electropherograms and not really knowing, what the process is. And so the samples are prepared in a 96 well plate or 384 well plate, depending on the throughput that's necessary. And again, like I mentioned, the hero of this is really the chip itself. And so I'm going to talk through a little bit just to give you an idea of how this functions. And so on the chip, we have these reservoirs that we kind of see outlined in these blue and red colors here. And these are prepared with gel and dye that we mix into the gel. So this is similar or analogous to if you were going to run an agarose gel where you prep the gel and you put your dye inside, and then the DNA or the RNA picks up that dye as it migrates through. We're essentially doing the same thing, but instead of doing it in a slab gel, it's happening in these capillaries. And so we can kind of see a cartoon representation here of these capillaries on the chip. And so first thing that happens is we apply a vacuum, we pull sample up into these capillaries, and they're mixed with our markers and our dye. And we migrate through some of these small capillaries before we make it to our separation channel. We see here on this next slide, we can pull a voltage across a few of these wells, and that begins to migrate our sample through this chip. And that long channel down the middle of the screen is our separation channel, where we're going to separate these nucleic acids by size. What we do is apply a very strong voltage between these top and bottom wells, and that actually pulls a very small plug of sample that's migrating through those other capillaries down the separation channel. And once we are in that separation channel, we're essentially doing electrophoresis just like you would do in a slab gel. We're separating based on size through that capillary and through that gel matrix. Now, towards the top, we see that circle, that detection window. On the instrument itself, we have laser. So we're doing laser-induced fluorescence. So as everything migrates through, it's excited by the laser, and then we have a detector on the instrument as well. And so that's what allows us to separate based on size, but then also get the electropherograms that we've been seeing today, right? Fluorescence versus time. And so, again, these are very small volumes, very small separation channels, right? So what's actually in the capillaries, we sip up picoliters, and what's actually separated down the capillaries is nanoliters worth of input. So that allows us to have very low limits of detection with our fluorescence system, and then also be very, very quick and boost that throughput. Now, what we care about on the instrument as well is not just that it's fast, but that the data we get is precise and looks good. And so I wanted to add one slide in here just to show some real world data that I've gathered while I'm traveling around the country working in labs with people. This is actually protein data that I'm showing as well. So I'll just highlight that briefly. And it's two very, very different samples. And what you're looking at is overlays of 5 replicates of these samples. On the left, we have what is a pretty crude lysate type sample. So a lot of gunk. We see a ton of these peaks in there. And then on the right, we see something that is a little bit more purified with that peak of interest over on the far right of that graph. And what we're looking at is that we want to be very precise when we gather data. So when we do multiple runs, we want to see that they overlay nicely, which they do on both of these. These are 5 replicates overlaid. And then we see on the left, we're looking at total concentration of the run. And we can get our means, our standard deviations, really tight CVs on this system, which is exactly what we want to get when we're trusting something to give us data, right? And then we see sizing. So we have the quantitation on that y-axis, looking at the area of these peaks. And then we have the actual sizing CVs as well, which we have on the right. And we see, again, really tight reproducible data, which is exactly what we want. And again, goes back to how that chip works that allows us to get this great reproducibility, but also great speed in doing so. With that all being said, I'm just going to highlight, one more quick study that's very similar to what we've seen before, just to kind of drive home this point. This is again back to using the lab chip for mRNA analysis. This is a publication from professionals at Merck, and this is a capillary electrophoresis method for looking at integrity and purity of mRNA in vaccines. So again, like we've heard, we all know the importance of this mRNA technology today, based on everything that's been going on recently in the world. But this is a quick method for evaluating these things. And what I like about this publication is if we read into this, a lot of the standard analytical parameters that are commonly used when developing these assays are included here. So we look at linearity, precision, and accuracy, as well as limits of detection and limits of quantitation. And what is concluded here is that this platform is especially useful for developing vaccines and looking at the integrity as these are, as we've seen before today from Kaio and his great talk as well. Just to show a little data from this as well, we've already seen very similar traces to this on the left, and we see this shown again on the top right, where our percent intact mRNA is shown to be stable in our -20 storage conditions. And then as we increase the temperature, it degrades as expected. And then what I really want to highlight from this, because Kyle, again, did a great job of showing very similar data to this early on, is, again, just to show on the instrument on the bottom right there, that as we load more RNA, right? So we have a concentration gradient across that x-axis there on the bottom. Our percent purity, or our total percent of that main peak, stays constant, which we want to see that our purity analysis is constant over concentration ranges. And then we also want to see that the area that we pick up is linear to the amount of RNA that we add, which is important, again, for back to the precision thing that we talked about, for trusting the data that we get out, right? We don't want to see different numbers if we load different amounts of RNA. And so we're very happy to see nice linear data like this as well. And so hopefully that gives a good background on kind of how the instrument works, what to expect when using it. And so with that, I'm going to be quiet. I'll go to the last slide and say thank you. Thank you, Kaio, for giving your talk, for all the information. It's awesome to hear how this is being used. And then thank you to everyone for attending. And I think that we would both love to take any questions that anybody may have.
Okay, great. Thanks to you both. We do have a few questions, and I guess we'll have to fight it out to answer them. But is there any effect of sample prep on the samples in the lab chip assay?
Do you want me to take this one, Kyle? I can do that.
Go ahead.
Yeah, there can be for sure. Anything that you might do during sample prep that could cause any sort of degradation, right? You know, if we're not being careful on the lab bench, can obviously shift. The amount that you load, one of the things that you will see is that we're working with very small volumes, so the amount of input you need is very little. So we're pipetting, a couple microliters in, and so we like to be very precise with our pipetting so that we're making sure we're not thinking we have more of one sample than another because we're pipetting these limited amounts. So it is possible that the actual sample prep could cause some changes, but it's all pretty physical, normal error that you would introduce in a normal workflow, if that makes sense.
Great. And does the lyophilized product have the same mRNA pattern as the solution form at 2, or does lyophilization product impact the mRNA as well? I'm not at TO. I'm not really sure.
Yeah, that's a good question. If the lyophilization process itself could induce some sort of degradation in the mRNA, we are actually also studying that now. So because that's a very valid question. So right now we are also sort of dismembering the lyophilization process into several steps, and then every step looking at how the mRNA looks after the freezing step, freezing 2, primary drying, secondary drying, and so on. Because yes, we don't know exactly how the lyophilization process is treating our mRNA. If those patterns are solely because of the molecules being in a powder form, or if that happened before during the process. So that's something that we are currently studying right now. So I don't really have the full answer to that. But yeah, we are investigating that.
Stay tuned.
Yeah.
Okay, well, speaking of another one you might not have the full answer to, is there a size limit to the mRNA molecules being analyzed by the technology, and has it been used with saRNA?
Yes. So each of these assays that we had kind of spoken of before has an upper limit. So the RNA goes up to, I believe, 6,000 base pairs off the top of my head is what we validate to. There is a little bit of range above that you'll get data on. But we don't necessarily validate that off of our ladders. So you kind of have to extrapolate that a little bit, and it's a little more qualitative than quantitative on that size range. And then can you remind what the second question was of that?
Yeah, has it been used with saRNA?
I believe so. We have an application note with silencing RNA or interfering RNAs. So yeah, any type of RNA will work theoretically on there, and I do believe that we have actually an application note on some interfering RNAs and small RNA. So we do have an assay as well that's a small RNA assay that's supposed to be only between up to about 100 base pairs. So yep.
Okay. I think this might have been a very specific part of the talk. Validated, does that equal GMP? And is it Part 11 compliant?
Yep. No, not much to say there. Yes, it's an additional package on the instrument, so you can get the instrument in both ways. You can get it with it or without, but it is 21 CFR Part 11 compliant for GMP facilities.
Okay, great. And where does the 96 well plate play a role in the separation step?
So the 96 well plate is just acting as essentially a holder for all the samples. So the 90, let me maybe, can they still see the slides as we talk? Okay, let me go back over here. So essentially right here on the side of the instrument, you see kind of a little diagram on the bottom there that looks kind of like a 96 well plate on that little black tray on the side. That's where the samples are loaded. And so the 96 well plate will be loaded there. It'll be pulled into the instrument with a robot. The chip is stationary, and then that robot moves the 96 well plate over for each sample. So if you have samples in column 1, right, it goes from 1 and then moves it up to 2 to 3 to 4 to 5 to 6 and so on and so forth. But once the samples are in the chip is where everything's happening. The 96 well plate is just reservoirs of sample for it to sip from.
Okay. This might be a little connected. So how many samples can be processed on a single chip? And are the chip wells reused? And if so, does this mean multiple pipetting steps per chip?
That is a good question. So the samples, so each chip is validated, right? So if we're talking DNA, RNA, these chips are validated out to 2,000 samples that we guarantee you should get 2,000 samples worth of lifetime out of the chip. The 400 number you see here is for our protein assays. Proteins are a little more gunky. They take a little bit of a harsher environment on that chip. Reusable as many times as you want. So you can do those 2,000 in one day. You can do the 2,000, 100 samples a day for 20 days. Do whatever you want. So they are reusable from that standpoint. Those wells on the chips, you can kind of see an image there, or maybe you can kind of see it a little bit better here. These we recommend being filled with fresh reagents each day, right? So there is multiple pipetting steps where we will mix the gel and the dye, these two tubes that we see on the screen up here in the reagent kit, and then those will be pipetted onto the chip. But it's not something such as that you need, once the chip is primed, you put enough gel into those reservoirs for the amount of samples you want to run. So if you're running a whole 384 well plate, you load the amount of gel necessary to run that much, put it in, and then you don't have to keep playing with the chip the rest of the day. You just let your samples run through.
Okay. And does the lab chip allow us to size mRNA?
Kyle, I'll let you take this one, man.
Well, you can estimate the size, yes. In my slides, I think you could see the size being estimated over the peak there. I just look at the questions here. If I compare it with another other techniques like LCMS, not LCMS, we just started with HPLC as well. But the thing is that I think the lab chip has better resolution. The other part of the question was, can you measure poly-A tail? So if you compare a poly-A tail, mRNA plus poly-A and mRNA without the poly-A, you could easily see that in the lab chip, actually. Don't really have a lot of experience with LCMS right now or HPLC to say about the comparison, but using the lab chip, yes, you can size the mRNA, and yes, you would be able to distinguish a poly-A tail containing mRNA and one that doesn't have one.
Okay, great. Thanks for reading the question there. And then I think the last question for the webcast, because we are running out of time. So you want to keep typing in questions. We will pass those on to Kaio, and the guys can follow up with you. But the last question is, can this be automated if there are more plates?
Yes. So there's a couple options for that. One, if people are using many plates, we have a little barcode reader that people like to do. So it's very common for people to have plates coming straight off liquid handlers, right? Doing very, very high throughput, having barcodes scanned, and then those are loaded right into the lab chip. Again, back to the chip prep, there's a certain amount of samples that can be loaded at once with one chip prep, right? So up to 384 samples. So if there's enough gel in those wells, you can do the 496 well plates back to back to back without having to play with the chip at all or do anything, right? So that can be very automated. And then we do have the ability to have a plate loader added onto the lab chip where it can load plate after plate into the system. That is something that has been done before. So it does have the ability to become very, very automated. Normally you would put that chip in there. After that 96 well plate or 384 well plate's done, you put a new plate in and move forward. But there is that possibility that we've seen before of having that whole process automated.
I'm on mute. So thanks all for joining us. And as a registered attendee, you'll receive a link to the recording of this webcast for review. And thanks to James and Kaio. And we look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye, guys.